Hi, hello everyone. Thank you, Professor Francisco Anquita, for your introduction. I want to start by thanking to the organizing committee for the kind invitation to participate in this session. It will be my greatest pleasure to take this opportunity to discuss with you very recent evidence on the role of non-coding RNAs in cardio-oncology, and in particular, their value as promising biomarkers for the early detection of cardiotoxicity. As you will know, the human genome is composed of protein-coding genes which represent only a very small proportion of the total genome, around 2 to 3%, and a vast proportion of genes that give rise to non-coding RNAs, which represent as much as 98% of the transcriptional output. The non-coding RNAs are usually divided on the basis of their size into small and long classes of non-coding RNAs. However, over the last decade, many different families have been recognized, and their functions on the cellular biology started to be identified. The main groups of non-coding RNAs described to be involved in cardiovascular physiology and disease are the microRNAs, long non-coding RNAs, and circular RNAs. MicroRNAs are small non-coding RNAs involved in the post-transcriptional regulation of gene expression. These small molecules, composed of 18 to 20 nucleotide sequences, predominant'ly exert their cellular function by interacting with complementary sequences in the 3' untranslated region of the target messenger RNA, thereby suppressing protein translation in a very targeted way through RNA interference mechanisms. It is important to recognize that microRNAs participate in a tissue-specific manner in very complex regulatory networks, which comprise different regulatory levels, multiple players, and multiple targets. For instance, each microRNA has the potential to target multiple different messenger RNAs. That means that a certain microRNA may inhibit different genes related to different metabolic pathways or inhibit a certain pathway at different levels, eventually producing a stronger biological effect. On top of that, different microRNAs may act simultaneously on the same messenger RNA target, producing a stronger translational repression and eventually a stronger biological effect. It has been shown that microRNAs modulate several critical biological processes, including cell proliferation, apoptosis, hypertrophy, and inflammation. And many studies had implicated the aberrant expression of microRNAs with differential down-expression or up-expression of specific microRNAs in the pathogenesis of many diseases, including cardiovascular diseases and cancers. Furthermore, it should be mentioned that in a context-dependent manner, the same microRNA could function as cardiomere, angiomere, or oncomere. The long non-coding RNAs are defined based on their dimensions. They are composed of more than 200 nucleotides, but they form a very heterogeneous group in the sense that some of them play their action in the nucleus, while others exert their functions at the cytosol in very different ways. Just to give you a number, estimated number of long non-coding RNAs in the human genome is 15,000, and they play very diverse functions, and their functions are just starting to be unveiled. It has been shown that some long non-coding RNAs act at the nucleus, exerting their biological functions through epigenetic modifications, transcriptional regulation, and modulating the localization and function of proteins, while others act at the cytosol as molecular sponges, titrating microRNAs away from their natural messenger RNA targets, in other words, fixing target microRNAs and proteins. That means that long non-coding RNAs may provide a counter-regulatory mechanism inhibiting specific microRNAs and preventing them of doing their inhibitory action at gene expression. Circular RNAs are the most recently discovered family of non-coding RNAs. Circular RNAs are covalently closed RNA rings formed through alternative back splicing of protein coding exons. They are involved in transcriptional and post-transcriptional regulation of gene expression, ranging their function from host gene regulation to scaffold and molecular sponge function. Indeed, it was recently discovered that circular RNAs might work as microRNA sponges binding to microRNAs and repressing their function. As microRNAs control a large set of biological processes, circular RNA sponge activity will also affect these biological pathways. Therefore, these various non-coding RNA families form a complex multi-layer regulatory network, and an intricate crosstalk exists between the several non-coding RNA species, fine-tuning the gene expression at transcriptional and post-transcriptional levels. In recent years, the roles of microRNAs, long non-coding RNAs, and circular RNAs in the pathophysiology of various diseases started to be established. In fact, it has become evident that the expression pattern of these molecules changes during disease progression, and aberrant down-expression or overexpression of specific species has been associated with the pathophysiology of cancer and cardiovascular diseases. Our group recently investigated the overall expression pattern of messenger RNAs, microRNAs, and circular RNAs in cardiac tissue samples collected in the left atrium of patients submitted to cardiac surgery, with history of paroxysmal atrial fibrillation, permanent atrial fibrillation, and a control group composed of patients without history of cardiac arrhythmia. We observed relevant changes of the overall expression pattern with the progression of the disease. The expression of some microRNAs was increased with the progression of the disease in parallel with the down expression of their predicted messenger RNA targets. But the dominant feature of AF was the down expression of numerous microRNAs. And by studying the expression pattern of circular RNAs, we started to reveal the crosstalk that exists between these two types of non-coding RNAs, between these two types of regulatory molecules during the progression of the disease. More precisely, we found that at the same time, microRNA expression decreases with the progression from paroxysmal to permanent AF, some abnormal circular RNAs started to be expressed in the atrial myocardium. And by analyzing their nucleotide sequence, we predicted that these aberrant circular RNAs might be acting precisely as sponges to the down-expressed microRNAs. In other words, the dysfunction of this regulatory network may be very important for the myocardial remodeling that occurs during AF progression. MicroRNA dysregulation in cancer was first reported back in 2002 when a cluster of two microRNAs, miR15 and miR16, was identified in a genomic region frequently deleted in chronic lymphocytic leukemia. This microRNA deletion was shown to act as, at least in part, through allowing higher expression of BCL2 oncogene, favoring this cancer progression. Since then, it has been documented that microRNAs have roles in all the landmarks in cancer biology, modulating tumor growth, invasion, angiogenesis, and immune response. Additionally, tumor microRNA profiles can define relevant subtypes, patient survival, and treatment response. As we will discuss further on, microRNAs can be also detected in the biological fluids, and extensive research is being conducted assessing the value of the quantification in the blood of some cancer-associated microRNAs as biomarkers to facilitate patient diagnosis, prognostic certification, monitoring, and treatment in the oncology field. The role of non-coding RNAs in cardio-oncology started to be unveiled very recently and is still a matter of great debate. As you all know, doxorubicin is widely used in clinical settings for a variety of malignancies, such as breast cancer, lymphomas, and leukemia. And this drug is also well known for its serious cardiotoxicity, with left ventricle dilatation, systolic dysfunction, and congestive heart failure, which impacts long-term outcomes. Several studies have suggested that apoptosis is the predominant cellular event in doxorubicin-induced cardiotoxicity. And in this setting, apoptosis may be the result of very different mechanisms and cellular pathways in which non-coding RNAs are critical players. For instance, doxorubicin-induced cardiotoxicity is closely associated with myocardial dysfunction, with excessive mitochondrial fission, loss of mitochondrial membrane permeability, and cytochrome C release. And you must remember that cardiomyocytes have high mitochondrial abundance for high energy production, and therefore these cells are extremely vulnerable to toxic agents that interfere on mitochondrial metabolism. Mitochondrial dysfunction results in excessive production of reactive oxygen species, inducing oxidative damage and resulting in tissue injury. Over the last 5 years, different microRNAs have been implicated in doxorubicin-induced cardiotoxicity. For instance, it has been observed that upon doxorubicin treatment, the expression of miR532 increases and the expression of miR499, which is a microRNA selectively expressed in cardiomyocytes, significantly decreases. This is relevant since miR532 was found to target ARC, an apoptosis suppressor, thus promoting mitochondrial fission, and miR-499 down expression leads to the upregulation of the target P21, a transcription factor involved in heart injury, subsequently causing mitochondrial dysfunction and apoptosis. And as you can see, many different microRNAs have been linked to the biological mechanisms implicated in doxorubicin-induced cardiotoxicity. Clearly, among the three types of non-coding RNAs, macroRNAs are the ones most extensively studied. The understanding of the roles of long coding RNAs and circular RNAs in doxorubicin-induced cardiotoxicity is still at an initial stage. However, mounting evidence suggests that both are associated with many cardiovascular diseases. The long non-coding RNA P21 was shown to play a regulatory role in doxorubicin-induced cardiomyopathy, and suppressing this long non-coding RNA was found to attenuate mitochondrial dysfunction, oxidative stress, and cardiomyocyte senescence. However, it should be highlighted that all these relevant observations came from in vitro studies. So precaution is required and further investigation is clearly needed, proving their participation in animal models and exploring their relevance in human disease. Only then it will be possible to consider the theoretical possibility of exploring the therapeutic manipulation of dysregulated non-coding RNAs to reverse or even to prevent chemotherapy-induced cardiotoxicity. Non-coding RNAs can be detected in extracellular medium and in several body fluids, including the blood, urine, and saliva. Their presence in the fluids is a natural process, not only related with cell death, but also with active secretion involving macroparticles, high-density lipoproteins, and exosomes. In fact, recent evidence suggests that non-coding RNAs may act as genetic hormones, being transported in the blood up to distant organs and modulating the gene expression in distant target cells. Incorporation of the non-coding RNAs in these extracellular carriers confers them great stability in the blood, which is a major requirement for a biomarker. The circulating non-coding RNAs present robust expression patterns and are expressed in a tissue-specific manner, which are also important features, making them very attractive biomarkers. Therefore, circulating microRNAs are being extensively explored as biomarkers for prognostic certification and drug efficacy assessment in different cancer types. And breast cancer seems to be the cancer under the most intense research. Just to give you an example, in this study, Wang and colleagues collected serum samples from almost 400 patients with advanced non-small cell lung cancer before treatment and found that the circulating levels of 17 microRNAs were able to identify the patients at the highest risk of death, and miR-16 exhibited the stronger association with outcome. Circulating microRNAs have also been the focus of great interest in the setting of cardiovascular disease. Back in 2010, D'Alessandro and colleagues found that some microRNAs typically expressed in cardiac tissue, MIR1, MIR133 A and B, 208B, and MIR499 rise in the bloodstream early in the temporal progression of myocardial infarction, and it was suggested that they could be valuable diagnostic biomarkers, even more sensitive and specific than, for instance, troponin levels. However, it should be mentioned that comparison at that time was made with the first generation of troponin tests, and that circulating microRNAs have lost interest when the comparison was done more recently, applying high sensitivity troponin assays as depicted in this graph. Therefore, the focus has moved from the diagnostic field to the prognostic stratification. Our group was deeply involved in exploring the value of circulating microRNAs as prognostic biomarkers in patients with a ST elevation acute myocardial infarction. We studied 142 patients in whom blood samples were taken at the time of urgent cardiac catheterization, and the circulating levels of various microRNAs were measured. We found an event-free survival significantly worse in patients with a higher MIR122-133B ratio, who had almost 9 times higher risk of death or myocardial infarction and 4 times higher risk of adverse cardiovascular events. Therefore, the MIR122-133B ratio is a very interesting biomarker for the early prognostic stratification of these patients with a very deadly condition. But the interest of circulating non-coding RNAs as clinical biomarkers extends to many other cardiovascular diseases. This diagram comes from another paper of our group in which we systematically reviewed the evidence relating circulating non-coding RNAs and cardiovascular diseases in terms of diagnosis, prognostic stratification, and also the evaluation of the treatment response. You can see how dense is this diagram, which means that some microRNAs are simultaneously related to various processes critical to cardiac remodeling and tissue healing, and therefore associated simultaneously to several cardiovascular diseases. In conclusion, it makes sense to say that a strong rationale exists to hypothesize that circulating RNAs may be relevant biomarkers for chemotherapy-induced cardiotoxicity. This study, conducted in Brazil, evaluated 56 female patients with breast cancer submitted to doxorubicin and they followed them during 1 year, carefully searching for cardiotoxicity. Three weeks after each cycle, blood samples were collected for troponin and microRNA quantification. Cardiotoxicity was observed in 18% of the population. The LV ejection fraction decreased in those 10 patients, but the final values were still in the normal range. As you can see in these graphs, the circulating levels of the cardiac-enriched microRNAs dynamically changed over time, and the magnitude of change was higher in the ones that later on developed cardiotoxicity. Among the several circulating microRNAs, MIR1 was identified as the best prognostic predictor. This cardiac-enriched microRNA presented high accuracy to detect patients that progressed with cardiotoxicity. And it was a significantly better predictor than serotonin and the 3 weeks after the second cycle. The MIR1 levels were already significantly higher in the women that suffered from cardiotoxicity 1 year after starting chemotherapy. Since then, other studies involving patients submitted to chemotherapy in the setting of different cancers, complemented by experimental evidence produced in animal models, reinforce the relevance of non-coding RNAs as biomarkers for the early detection of cardiotoxicity. Up to now, the most promising ones are the cardiac-enriched microRNAs, namely MIR1, 133A and B, and 499, and the profibrotic microRNA 29 family, which has been previously related to atrial fibrosis in the setting of atrial fibrillation and to heart failure in the setting of ischemic heart disease. And it also seems that it is a relevant player in the setting of chemotherapy-induced cardiotoxicity. So, in conclusion, it is extremely important to understand the mechanisms of chemotherapy-induced cardiotoxicity, to develop methods for the early detection of cytotoxicity and therapeutic applications. MicroRNAs, long non-coding RNAs, and circular RNAs have been shown to have significant functions in the cardiovascular system and are likely to be also relevant in the pathophysiology of cardiotoxicity. Non-coding RNAs are promising biomarkers, but further studies are clearly needed to clarify their value for the timely detection of chemotherapy-induced cardiotoxicity. Thank you very much.